Hey everyone, and welcome to this high-yield explainer. Look, if you're gearing up for board exams or you just really want to bulletproof your clinical knowledge, you are in exactly the right place. Today, we are tackling enteric pathogens and food poisoning. And let me tell you right now, mastering the specific mechanisms of these gut bugs, it is the ultimate exam cheat code. Because once you actually understand the why behind the symptoms, you will literally never have to brute force memorize a microbiology textbook again.
Okay, let's dive right into this. Here's our roadmap for today. We'll hit pathogenic mechanisms, host defenses, the legendary food poisoning table, a few crucial exam pearls, the physiology of why ORS works, and finally, lab diagnosis.
All right, first up, Section 1, Pathogenic Mechanisms.
So, transitioning from just knowing a laundry list of bacteria names to understanding how these bugs actually launch their attacks, that makes anticipating their clinical symptoms infinitely easier. Now, you might be wondering about inoculum size, which, by the way, is a heavily tested concept. Just look at the contrast. We've got organisms with a terrifyingly low infective dose. I mean, we're talking just 10 to 100 organisms for things like Shigella, EHEC, and Giardia. Because it takes so little to cause an infection, these spread super easily from person to person. But contrast that with Vibrio cholerae. Cholerae requires a massive dose, 100,000 to 100 million organisms, just to survive the acid in your stomach. And that directly dictates that cholera isn't going to easily spread by a simple handshake, right? It absolutely requires a heavily contaminated water or food supply.
Now, beyond just inoculum size, these bugs generally rely on three other main virulent strategies: adherence, toxin production, and invasion. Some bugs, like EPEC and EHEC, literally adhere to and destroy your intestinal brush borders. But the toxins, that is where the test writers are really going to try to trip you up. Enterotoxins basically ramp up CMP or cGMP, and that causes massive watery diarrhea, but usually without a fever. Cytotoxins, on the other hand, are like microscopic wrecking balls. They physically destroy the mucosal cells, which leads directly to bloody dysentery and inflammation. And neurotoxins? Yeah, those are preformed in the food itself, acting right on your central nervous system to cause really rapid, violent vomiting. Finally, you have invasion. Bugs like Salmonella and Shigella that physically invade deep into the tissue.
Moving right along to Section 2, Host Defenses.
Okay, so with all these sophisticated attack strategies, how are we not just constantly sick? Seriously, take a second and think about it. You encounter countless bacteria every single day. So what's actually stopping these pathogens right now in your own body? Well, we actually have a remarkably formidable biological shield. First up, gastric acid. This is a massive barrier. So red flag time. If you have a patient who's on antacids or PPIs, or maybe they've had a gastrectomy, that is a huge warning sign. Their hypochlorhydria severely increases their risk for cholera and salmonella. Next is intestinal motility. Normal peristalsis literally just flushes the bugs right out. Then we have intestinal mucin and secretory IgA, which are basically our mucosal first responders. And finally, our normal intestinal microbiota provides something called colonization resistance. Here's another clinical red flag for the boards. If you give antibiotics, you disrupt that normal flora. You're basically rolling out the red carpet for C. diff to take over the whole shebang.
All right, Section 3, and we have reached the holy grail of this explainer, the food poisoning table.
We usually break food poisoning down by incubation time. Notice the short incubation group, that classic 1 to 6 hour window. The exam pattern here is absolutely unmistakable. If the question stem says a patient is violently ill just a few hours after eating potato salad at a summer picnic, bam, it's Staph aureus. If it happens after eating reheated fried rice, that's the emetic form of Bacillus cereus. And why does it happen so fast? Because the neurotoxin was already preformed before they even took a single bite.
Contrast that short window with the longer incubation group, which takes 1 to 4 days, simply because the bacteria actually need time to colonize, multiply, or invade. Now, multiple choice questions love to make you differentiate within this specific group. For example, if a patient has bloody diarrhea, immediately check their temperature. Shigella presents with bloody diarrhea and a high fever because it invades, but EHEC, which you'll often see from undercooked ground beef, presents with bloody diarrhea but low or no fever because it's entirely toxin-mediated.
Section 4, Exam Pearls. Pay very close attention here.
I need you to use your most authoritative clinical voice in your head right now because this is an absolute non-negotiable rule. Do not give antibiotics to a patient with EHEC. That's E. coli 0157H7. Why? Because giving antibiotics induces Shiga toxin phage replication. Basically, it means the dying bacteria release a massive wave of toxins, and that increases the risk of hemolytic uremic syndrome, which is a potentially fatal kidney failure, by 20-fold. No antibiotics for EHEC.
And this brilliantly illustrates another classic exam trap. One single bug, two entirely different clinical presentations. Bacillus cereus can cause an emetic form, 1 to 6 hours, preformed toxin, severe vomiting, usually from fried rice, or it can cause a diarrheal form, 8 to 16 hours, driven by an enterotoxin produced inside the gut after eating contaminated meat or veggies, causing diarrhea with almost no vomiting. The food source and the symptom timeline, they literally hand you the exact answer.
Let's quickly rapid-fire the other antibiotic rules you absolutely have to know. We just said a hard no to EHEC. For Salmonella, routine antibiotics actually prolong colonization, so we really only give them to high-risk patients. For Shigella, absolutely give them. It significantly shortens the severe illness. And for Campylobacter, because there's widespread fluoroquinolone resistance out there, especially in Asia, you must reach for a macrolide instead, like azithromycin.
Next up, Section 5, Why ORS Works.
We are about to look at one of the most brilliant physiological workarounds in the entire history of medicine. Let's see how this builds step by step. Step 1, the cholera toxin basically locks adenylate cyclase in the on position. That drives up CAMP, which forces massive chloride and water secretion into the gut. The patient literally pours out fluids. But here is the absolute genius of Step 2. The SGLT1 transporter in the small intestine, it is completely unaffected by the cholera toxin. So Step 3, if you give a patient a specific, calculated ratio of glucose and sodium together, they co-transport across the membrane. And as they do, they drag water back into the body with them, completely bypassing the cholera toxin's blockade. This incredibly simple solution, literally just the right mix of sugar, salt, and water, is nothing short of miraculous. To put that into perspective, before ORS, untreated cholera had a mortality rate of greater than 50%. 50! By utilizing that physiological backdoor, ORS dropped cholera mortality down to less than 1%. It's a game changer that has saved tens of millions of lives.
Finally, Section 6, Lab Diagnosis.
Okay, so we know the bugs, but how do we definitively prove it in the lab? Here are your high-yield exam associations. Fecal lactoferrin is highly sensitive for invasive inflammation. For cultures, remember that EHEC is a sorbitol non-fermenter, so it shows up as white or colorless colonies on a sorbitol MacConkey agar, very testable. Salmonella and Shigella are both non-lactose fermenters, and to grow Campylobacter, you need a specialized microaerophilic environment heated to exactly 42 degrees Celsius.
I'll wrap up this explainer by leaving you with this thought. Today, we have these amazing rapid multiplex PCR panels that can detect pathogen DNA instantly. Yet, clinical guidelines still insist we follow up with old-school stool cultures. Why? Well, because while a PCR gives you a name, a live culture is the only way to track antimicrobial resistance patterns and perform the public health typing we need to stop outbreaks at their actual source. The battle just doesn't end with a diagnosis, It extends right into population health. Keep questioning the why behind these guidelines, keep reviewing these pathways, and I'll see you in the next explainer.